Mhm.
Welcome, folks online. Aloha. Slice of PI-CASC, hosted by the Pacific Islands Climate Adaptation Science Center. Hopefully you're hearing us okay now online. This Slice of PI-CASC seminar series provides a venue for sharing state-of-the-art climate adaptation research and science management applications in Hawaii, the U.S. affiliated Pacific Islands, and beyond. I'm Brad Romine, the Consortium Deputy Director of the Pacific Islands Climate Adaptation Science Center. This presentation is being recorded and will be available on our website under News and Events on the PI-CASC website. So please share it around for those that couldn't make it, or if you want to check it out again, you'll find it there. For those of you joining us online, please keep your microphones muted. We will have time for questions and answers at the end of the presentations. Also, feel free to leave your questions along the way in the chat, or we can have you chime in afterwards. So today, we're very excited to have 4 speakers joining us for this Slice of PI-CASC seminar. And we're going to hear about ecological and sociocultural response to transplanting corals to enhance reef resilience. I'm going to introduce our folks, I think, in speaking order here. First of all, we have Kira Hughes, the Managing Director with the University of Hawaii Institute of Marine Biology Coral Resilience Lab. Along with Kira, we also have Kinohi Fukumitsu with Kekula Nui o Waimanalu is going to talk about some of the community engagement that this project has done. And I see Kinohi online. Thanks for joining us, Kinohi. Aloha. Then we also have Jasmine Chang, who's a research assistant with HIMB and the Coral Resilience Lab. And last but not least, Dr. Crawford Drury is going to talk to us about some of the work also from who's assistant researcher with the Coral Resilience Lab. So with that, join me in welcoming the team from HIMB, and I'll hand it over to Kira. We'll get started.
All right, sounds good. Okay, can everyone hear me okay online? We're good? Okay, great. So the 4 of us are presenting, but this is actually a much larger project with a lot of people. And so we try to list a lot of the key people here. But I wanted to give a special shout out to Kirk, Ilima, and Puanani, who have been really integral to making this project happen. So first of all, just talking about the general importance of reefs. They're just majestic, right? Like people love to go diving because they're just gorgeous and mesmerizing. And they're also really complex. They're actually animals. And every time we think we figure something out about them, there's something more to learn. They're really important here in Hawaii because they are our main coastal defenders. They provide habitat for all sorts of organisms, especially fish and fisheries are dependent on them, provide food for communities, and they're a main part of our economy. However, in Hawaii, there's an added importance of Hawaiian relationships with koa or coral. There's a creation chant called the Kumulipo, and that shows the connection of Hawaiians deeply, ancestrally, and spiritually, because the coral polyps are one of the first to emerge as life. And before Western influence here in Hawaii, Hawaiians were able to foster sustainable use of these types of resources. So there have been a lot of impacts, human-induced, on corals over the years, but the biggest threat right now is warm water that is causing corals to really stress and causing them to lose their symbiotic algae symbionts and turn bright white or bleach. So this is really dangerous because they can recover if the stress goes away, but if that doesn't happen, then they usually die. We've seen this here in Hawaii during the 2014 and 15 back-to-back bleaching events was the most extreme. And during that time, we observed some corals from the same species side by side, ones that bleached and the other one did not. So we started to investigate the ones that did not bleach, and we've come to find that they are much more thermally tolerant, and therefore they're much more likely to survive into the future because these coral bleaching events are anticipated to last longer and be more extreme in the coming years. So we started a restoration project called Restore with Resilience, where we found corals that were detached from the main reef and just took a little piece, a biopsy, and sampled them to test their thermal tolerance, and then selectively outplanted the ones that were more thermally tolerant. And this was done across 3 sites on Oahu, 1 on Maui, and we were able to integrate the community with that project. So this is just like a first step to looking at how we can prepare our reefs for the future. We worked within the same location for that project. So any corals that came that we wanted to outplant in a bay, say Kaneohe Bay, we worked with corals from that bay. But really, when you're thinking about big impacts to prepare corals for the future, in restoration, there's a concept called transplantation, where you can take corals from one place, maybe a warmer site, like Kaneohe Bay, which is about 1 degree Celsius or 3 degrees Fahrenheit warmer than, say, Waimanalo. That is open ocean and has kind of cooler, more movement going on out there. And if it gets too hot for corals in that bay, it's the largest sheltered bay in all of Hawaii, then maybe you can move those to a place like Waimanalo where it's nice and cool to be able to save those corals. None of this has ever been done before in Hawaii. So it's really, figuring out like, can corals even be moved to different places? Can they even, if they are thermally tolerant already, can they retain that? So those were some of the questions that we had going into this project. One of the reasons we were really interested in Waimanalo is that our colleagues had collected corals from around the whole island of Oahu, and Waimanalo had the least thermally tolerant corals. So they were most susceptible to heat stress, whereas Kaneohe had the the strongest corals for heat tolerance. Now, I grew up in Kailua, which is next door to Waimanalo. So I'm pretty aware that people in Waimanalo take care of their own resources, and they're very protective of their resources. And it's great. So they have these community-based resource management programs there. This program is an example. It's been running for 7 years. It's the Waimanalo Limuhui. Kinohi is going to talk a little bit about it next because it's part of this organization that we partnered with. But really, we were able to kind of go to some of these ongoing projects and say like, hey, we're interested in this kind of biological, physiological project. But then we also need to know how how your community feels about that. So does Waimanalo even want other people helping to protect their marine resources? And if so, what does that look like? And then really thinking about these coral interventions. So, working within the same bay and taking corals that were already dislodged from reefs seems pretty minimal. But this transplantation is kind of a next step, and that's an intervention. So there's much more extreme interventions, like you can change out genes, you can do all sorts of things to try to enhance thermal tolerance. So these are still, relatively risk-free interventions, but really like, where is that threshold? What are people actually comfortable with at this stage? And with that, I'll pass it off to Kinohi to talk about Ke Kuanui o Waimanalo.
Thank you, Kira. Like Kira said, my name is Kinohi Fukumitsu. I'm from Waimanalo. I actually never moved from Waimanalo, so I'm a lifetime citizen of this community, which gives me like great and different types of perspectives. Growing up, I actually went to school at Kamehameha schools, so it created kind of like a situation where I could be outside of my community for a majority of the day and kind of look back in. So it gave me a different type of perspective within my community. And then now that, a lot of my teachings and learnings has been based in a fish pond in He'eia for 12 years. So a lot of my teachings and learnings and kilo actually come from He'eia. And my transition to Waimanalo has been slow and steady, but that's kind of what I'm speaking from. So when it comes to Ke Kula Nui o Waimanalo, my involvement, I would say I have a unique perspective of being a haumana of this school, because if you look up what Ke Kulanui means, it's actually a college. So um, through Ke Kulanui o Amanalo, me and my family have been able to take a bunch of courses that they offer, which has a lot to do with food sustainability. So one you guys just got introduced to is Wamanalo Limuhui. It's one of our well-known programs, but it's to restore the abundance of native seaweeds in Waimanalo. Another one that I'm passionate about is the restoration of Pahonu fish pond. Because of my background in fish ponds, that's kind of where I have dwelt. Um, and that's kind of my niche. But I've been really enjoying learning the science um, to kind of support my learning of fish ponds as well through these programs. Kilo and monitoring is another thing that we do, especially we've been fortunate enough to start this new Kilo Kamakai program for our Kiki and Waimanalo as well. And basically, this is our vision and mission and our location and our logo. And there's so much that we do that I probably could take up the whole hour speaking of this organization that I'm involved with and have the honor of learning from. So we can go to the next slide, but that's the hat that I wear in my community. I'm still learning. So you see these pictures um, of some of our programming that we offer. As you can see, we do aquaponics, giving native Hawaiian families the opportunity to grow their own food. Um, we also have a chicken program where people can grow their own food through chickens and eggs. We also have taught our children how to harvest chickens as well. But yeah, like it's a very holistic or community-based organization, and there's so many facets to it. But we can go to the next slide. But these three are the main drivers of all of the programming at Ke Kulanui o Waimanalo. Limu, Aipono, and La'au. So native seaweeds, eating healthy foods, and creating our own medicine. Throughout all of the programs, these are the main three basis of our programs and why we do all that we do. Our next level, I would say, because I'm speaking as a Haumana, I'm able to actually now teach my children a lot of the things that I learned from the programs at Ke Kulanui o Amanalo. So that succession exists, and that succession is part of that main driver. And I had to express that point before we go on, because I feel like that is why we create the programs that we do. Mahalo, Kira.
Hi, Kinohi. All right, so this has been a 4-year project, and I thought I'd share a little bit of a timeline to give you a better idea of how everything evolved. So back during COVID times, we applied for this grant, and we were able to gain letters of support from Ke Kulanui o Waimanalo and Waimanalo Canoe Club to work with that community, which I was really surprised. I didn't expect them to be willing to right away support a project that seemed a little bit more extreme, but the real reasoning behind it was that people in Waimanalo just wanted to learn a little bit more about what we were doing and see what we could make happen together. So that's the first step. As soon as we decided that they were going to support us in this application, we started building pilina or relationships right away. Before we knew we received funding, anything like that, whether or not we knew we were going to, we just knew that we wanted to build those relationships. So I started by meeting with the board of directors with Kekulanui o Waimanalo. I gave a virtual presentation to one of their programs, which is called the Waimanalo Pono Research Hui. So they set up this hui to be able to review as a community any research projects that want to come into Waimanalo. So typically what the people they hear from is like people that want to study health of Hawaiians or something similar to that. Whereas this project was pretty different to them. It was the first project to, one of the first projects to really ask about coral reefs and what they wanted to do for the management there. So I gave that kind of initial presentation, and then this group has something they call the shark tank. And it really is sometimes that vicious, where you go up and you present, and then usually afterwards the community votes about whether they want you to proceed or not. So I dipped into that shark tank for their very first larger group meeting after COVID. I was still mainly wearing a mask except for this picture. At least, you know, it was outdoors, but it was the first time meeting face to face. So we'd had these virtual meetings. We were trying to build these relationships online. As you guys all have learned through COVID, it's very difficult to do. So I gave that Shark Tank presentation, and because of what, you know, Kinohi's history in Heia, and because of the Limu project that they have in Waimanalo, they're very aware of concerns with aquatic invasive species. So the big concern is like, okay, if you're bringing coral fragments over from Kaneohe Bay, which is known to be a disaster for invasive algae, are you going to be bringing accidentally that algae over to Waimanalo? So that was a big concern, and totally valid concern, right? Like, we work on corals all the time. We know what it looks like. We know what it looks like when we fragment them, how clean we can make them. But, and we wouldn't want to disrupt the system in Waimanalo. But I said, totally valid. Let's look at how we can make the community feel a little bit better about this. So it took the time to really do a lot of research on that. And in the meantime, we just kept building pilina. So they came to some of our Restore with Resilience events, which are called Hanapukoa. Our lab came to the Pohonu Pond that Kinohi mentioned. We were doing outreach in the elementary schools in Waimanalo. And then finally, we received the funding, which is great. Okay, so 2022 was really the big year that, you know, COVID was a little bit less of an issue, and we were really able to kind of get going. And so we knew that the Waimanalo community was interested in working with us somehow. We just weren't sure how yet. And one of the big pieces of feedback was like, we really want people from Waimanalo leading these projects. And that's what we wanted to. That's what we put into the application, was that we wanted to recruit people from Waimanalo to work on this project. However, you know, trying to get someone geographic specific is sometimes harder than that sounds. But luckily, we were able to find Jazz, who's here today to present, and also Pua, who I believe is online. And they were both undergraduate students at UH, and they were able to join us at the lab. And we were able to help kind of build them up to be able to lead some kind of project there with their community. So we went back into the Shark Tank with them. I was like, you guys are the ones that should be doing this, right? Like, this is your community. And kind of, well, sorry, let me back up a second. So with I went back into the Shark Tank with them on board, and we said, okay, we're going to have people from Waimanalo on the project, and here's a plan for the invasive species. So we talked with Hawaii Pacific University that has Oceanic Institute over there, and we said, can we pay to set up a quarantine system at your site? And then when we bring corals over, we can have at least a 30-day quarantine. We can inspect them. We can have Dr. Cecilia Smith and Uncle Wally come and inspect them as well, make sure everything's good. The system went into the ground. It didn't go out into the ocean. So pretty much tried to make the community as comfortable as possible. And at the end of that meeting, the answer was still no. So they didn't know us well enough at that point, and it was still a risk. And they didn't know enough about the coral reefs to really be able to make that decision on behalf of the rest of their community. So we completely respected that, and we pivoted. So with that is the point when Jazz and Pua were able to come up with another idea in order to kind of work with the community and share more about these reefs with them. At the same time, we were able to obtain a permit to be able to do the work because we had already built that relationship with Department of Aquatic Resources, that they trusted us to be able to do this because they know our work. But that will become important later. So for the transplant, we said, okay, well, how about if we go to a different community? And they said, we have no say over other communities. That's up to those communities. So we said, there's still a lot of information you can gain from going over to Kailua Bay. And it's a lot safer because you're just going around the corner where a lot of that invasive algae is probably coming around and transferring anyway. And so we consulted with Ko'olaupoku Hawaiian Civic Club. And at the same time, we were receiving another project that was a hybrid natural artificial reef that would be in that space. And but we had never transferred corals. Corals would be coming from our labs in Kaneohe Bay. So this would actually inform this other project that's sponsored by DARPA to be able to inform that first before ever putting any corals out there and making sure that they would survive. And we were able to revise that permit to work in Kailua Bay instead. So during that time, once we figured all of this out, we were able to deepen pilina. We had some awesome overnight weekend workshops with 50 members from the Waimanalo-Pono Research Hui. We did activities. We transferred activities, some research, and some of the activities presented from the community. We did a spring break keiki visit. We constantly attended the monthly meetings and went to Lima Hui events when we could. And that led us to co-develop a project together. Rather than us coming in and saying what we wanted to do, we were able to co-develop something together. So Jazz is going to talk about the 3D maps. We talked about a data ownership, and that was really important when it came up. But we decided, since we were working so closely together, that we would just check with each other the entire time. So we drafted an MOU, and we were like, maybe this isn't necessary. And then Kinohi will talk about the Kamakai Ola Waimanalo program that we were able to create for the keiki. That was mainly on the Kekulenui Waimanalo side. And in the meantime, we're still working on this transplant project in Kailua. So we're able to do those collections in 2023. So this year, we had a second overnight workshop early in the year because we had such a great time last time. We really recognized how spending that much time together helped. We deployed that transplant in Kailua. We hosted the first ever Wahine Research Summit, which is something totally put on by Waimanalo-Pono Research Hui. We were just lucky enough to be involved and to host them. But it was other people that were interested in this concept of Pono Research Hui and how can they bring that to their communities statewide. How can they look for resources? How can they give their community more of a voice? So these were all female Hawaiian leaders from across the state. And moving forward, PUA is working on some curriculum, and Jazz is going to assist Kirk with some kapuna interviews in the future. And that's back to Kinohi.
Thanks, Kira. So I can't show up today without bringing my keiki along with us. This is actually a picture of our first initial meeting in March of this year, 2024. And in this program, thanks to some BWET funding or watershed funding, we were able to create a program to teach our keiki both Western science and kilo and Hawaiian kilo. And we teach the kids, we're trying to teach the kids to be fluent in both languages, basically. That's the easiest way that I can explain it to you. But what we were noticing as a gap in our community was baseline data in our four sites in Waimanalo. And we chose Muliwaiolana, which is at the beach park. We chose Kaiona Beach Park, which is where our local is. And then we have Kaupo Bay, which is Makapu'u. And then we also include Avava Malu, which is near Sandy Beach, or what is known as Allen Davis now. So those four sites, we go out every Monday and we take the kids with whoever's keiki shows up. We do our normal kilo routine, which is taking wind measurements and salinity measurements, and we also have them observe things in their natural environment. They shape their own observation sheets, and they are in charge of their own research and data. So it's been very exciting to see what they're observing when most people would think that they're not doing anything at all productive. They're actually making a lot of observations without us knowing a lot of the time. So figuring out ways to assess those types of measurements has been one of our goals, and measuring confidence and capacity within our children has been a blessing for us to be able to run these types of programs. Another big goal for us is to have our Keiki be the researchers. So thanks to people like Kira who come in with, it, like she said, it's not an easy community to engage with, to ask, to spend time with. And we know it. But it's very important. So I appreciate that Kira really drives that in as like important for our community of Wamanalo. But we really want to see in the future a whole bunch of researchers that we can tap and look to within our community. And the only way that we found that we can help with that is creating more scientists. So that's our mission with this program. Thanks, Kira.
Sorry.
No.
Hello, Kinohi. Aloha mai kako. My name is Jasmine, or they call me Jazz as well. I am an undergraduate here at UH. I'm studying marine biology and Hawaiian language, and I'm also a part of the Coral Resilience Lab as a lab technician, research assistant, loose terms. I'm gonna kind of switch and talk a little bit more about Western science and what me and my colleague Puanani have been working on. So I'm going to be talking briefly about photogrammetry, which is a newer methodology that's commonly used in our lab. It includes the photographing of reef ecosystems, followed by the use of Idasoft Metashape Professional Photogrammetry software. It's a mouthful. And we use this technology to create 3D orthomosaics of reefs. And from these models, which we call 3D orthomosaics, we can pull things like morphometric, what's the word for that? Morphometric analysis from these models, as well as biodiversity assessments and various other types of data. They're very useful. And this photo is just a small example of an orthomosaic that me and Pua made from a reef in Kaneohe Bay. So training members of Waimanalo community. My colleague Pua and I were trained in photogrammetry by Spencer Miller in Kaneohe Bay. We learned everything from driving the boat into the bay, photographing the reef, all the way to actually developing and processing these photos into 3D orthomosaics using the Agisoft software. Yeah, and this is really important. I think the big thing is that me and Pua are both born and raised in Waimanalo. We're both undergraduates pursuing our degree at UH right now. And we're both Native Hawaiian members of the Waimanalo community. And it's important that we learn these specific skill sets in our community so we can keep up with Western science practices. Yes. And it's important that we keep up so that we can participate in our community with these practices through the career that we want to continue down. And going off of this, Pua and I have also been making efforts to implement Indigenous Hawaiian knowledge into Western science. Specifically, Pua is working on a curriculum to teach keiki how to do photogrammetry by tying in the practices of hula and oli. and the way that we view modern day palapala aina or maps, in this case, reef maps, which is really cool. And lastly, right before this keynote, he was sharing a little bit about the Kamakaiola Waimanalo program, which I have been able to participate in. And I've been able to work with the keiki in our community, and it's just been an amazing experience spending time with these remarkable kids and watching them build pilina or relationship with their aina hanau, their birthplace, and giving them a space to learn more about Western science, which is a foreign idea to a lot of people, but through the lens of Kanaka, which has been amazing. And overall, we have been working to understand both sides of science, hoping to potentially build a bridge between the two. And the most enriching thing that I've taken away from all of this project as a whole, is the importance of implementing community into science, or more so the need for science to be more considerate of community when we propose these studies, in order to ensure that these studies are actually culturally appropriate and relevant to the people who live there, depend on, and have deep connections to these places. Thank you. Oh, sorry.
Hi, everybody. I'm Ford, bringing up the rear here. I am originally from Georgia, and I grew up in Florida, and I moved to Hawaii about 7 years ago, and I've lived in Kaneohe since. I just wanted to thank Kinohi and Jazz and Pua and Kira for kind of the context they provided today, because before I moved here, there's never any consideration of what Jazz just mentioned, which is like, to be considerate of the community as you're working in your research. And this has been a growing and learning experience for me too, and one that I'm really appreciative of. I'm going to switch gears a little bit and talk about this, the coral side of our project with this experiment that we are fondly calling K2K for Kailua to Kaneohe, or Kaneohe to Kailua. I'd like to give a shout out to Ali Klein, who hopefully is on Zoom here, who is a graduate student at FAU and has been working on this project as part of the larger collaboration that Kira mentioned with this hybrid reef structure. So way back at the beginning of the talk, Kira mentioned a couple of the types of questions that we want to be able to ask using this research. And they focus primarily on like management and conservation, being able to do it responsibly and understanding what the consequences are. And so one relatively, I think, conservative approach is the idea that we might be able to take corals from a place that is warmer and maybe a little more stressful and move them into a nearby climate refugia, thereby buying time. And so we have a perfect ecosystem where we can try to address this question using the different bays on the windward side of Oahu. But in order to use that as an approach to protect our coral reefs in Hawaii, we need to understand a lot more about the fundamentals of where thermal tolerance, where this climate robustness actually comes from. Is it a characteristic of the corals themselves, the reef they come from, the bay they come from, the species? Where do we find, what do we find are the drivers of these traits? And so we're trying to address a number of these questions, and most importantly, understand if the characteristics of corals that we care about, for example, climate resilience, are conserved after we move them to a different place. And so Kira mentioned that we pivoted to a transplant between Kaneohe Bay and Kailua Bay. Just realized that I can... Kanohi Bay and Kailua Bay at Ulupa'u. And Kanohi Bay is the largest sheltered embayment in Hawaii. It's got really high coral cover. It's kind of one of the international gold standards for what you can do if you actually try to remove the stress on an ecosystem and let it recover. 30, 40 years ago, it was an algal-dominated state. And when pollution and dredging and kind of wastewater were removed, it was actually able to recover and is now what we consider to be a really, nice ecosystem with high coral cover. But it also got somewhat warmer temperatures and lower pH than the offshore pelagic waters. And so that means the corals in Kaneohe Bay today are experiencing environmental conditions that are more similar to what corals elsewhere in Hawaii will be experiencing in the future. And so in some ways, we can think of this as them already being somewhat pre-adapted for climate change. And then our other site is Ulupa'u. It's got higher wave action. It's exposed to much more pelagic water, which gives it lower temperatures, and is a kind of more similar to the other offshore reefs around the state of Hawaii. And so we wanted to move some corals back and forth between these two places to understand how we might be able to use that for conservation and management. So here's a map of our study system, and we collected corals, both Montsipora capitata and Parietes compressa, from 3 sites with the blue arrows here. And we chose these 3 sites because they are the warmest, coldest, and most average temperatures of this kind of long-term temperature record that we have from 30 sites throughout Kinohi Bay. And we thought that perhaps by tailoring our collections to these environmental conditions, we might get an additional signal of the influence of the environment. We also collected corals from Kailua, brought them all back to the lab and quarantined them for a while. And then they were outplanted to 2 sites with these little black arrows here. The blue site is nursery tables run by the Coral Resilience Lab at Reef 13. And then this pink site is the same location as this hybrid reef that Kira mentioned before, and the same location from which these corals were originally collected. So we chose these 2 species because they are common throughout the state. They're dominant in Hawaii. They also represent 2 kind of different reproductive patterns. Montsipora is cosmopolitan. It's found all over the Pacific, but Parietes is found primarily in the main Hawaiian Islands. And so we collected 20 genotypes of each of these species, brought them back to the lab, cut them up with a bandsaw, and mounted them in these kind of little thumb-sized fragments. And so this gives us the ability to ask questions about the site and the bay the corals came from, as well as the coral genotype, so the individual colony, because we know that has a lot of effect on the different traits and phenotypes of corals. And then obviously the 2 different species. But we had not outplanted corals in this on these plugs before, and so this was a bit of a step forward in methods development too for how to do restoration work using corals in Hawaii. I think one important point on that note is that branching corals in particular benefit from this kind of idea of pruning vigor. So when you fragment them, they grow more quickly. And so propagating corals in this way is one way that you can take a larger colony and generate a lot more biomass and a healthier ecosystem through restoration over time. We made the initial collections in November of 2023 and mounted them up, but then we wanted them to recover. We also needed to quarantine them, so we kept them for about 5 months and then got them back out to our Common Garden Nursery in Kinohi Bay. We collected baseline data before outplanting these corals from an additional fragment that wasn't going out. And then we outplanted them at the beginning of this past summer. We're still monitoring them. The last monitoring time point was in October, I guess. And we're planning to continue that for as long as is feasible, but at least through hopefully this time next year, where we may or may not see another natural bleaching event, which typically occurs around October in Hawaii. So a lot of the questions we want to ask rely on the physiology and phenotypes of these corals, where their drivers of those traits come from and how well they're conserved. And so the primary things that we're measuring are kind of both ecological and molecular. So this little doohickey here is a custom photogrammetry rig. Jazz mentioned 3D photogrammetry. You can do that at the scale of reefs, but we can also do it at the scale of these thumb-sized corals. And it's basically a rotating turntable with a bunch of cameras. And we can build these really precise 3D models of these corals. And that enables us to look at shape and complexity, which really influence the function of coral reefs. The coral structure is what creates the function of a reef. And we couple that with buoyant weights. We can also look at the skeletal calcification dynamics of these corals. And then lastly, we used this imaging PAM fluorometer, which is basically just a really fancy light reading instrument that allows us to measure the health of the photosynthesis of the coral. And this is our primary metric for understanding bleaching, because as that photosynthetic health declines, corals are also bleaching and eventually will die if they don't recover. We also wanted to measure a couple of molecular phenotypes that I'm not going to talk about too much today, but we've collected samples for metabolomics and transcriptomics so we can understand gene expression and biochemistry and how those are kind of these intermediate traits that may change when we start to move corals around different environments. So one of the primary questions we have are the relative thermal tolerance, the relative climate robustness of the individual corals conserved after we move them. And so that meant that we needed to assay these corals before they went out. So we had all these fragments that were going to be transplanted. We had an extra piece that we kept in the lab, and we exposed it to this temperature stress. So it's a little hard to see here, but this goes up to about 31.5 degrees Celsius, which is quite hot. That's about 1.5 degrees hotter than your summertime max in Kinohi Bay for about 2 weeks. And then we measured these corals throughout this experiment to generate these decay curves. And so on the y-axis here, you basically have the health of the photosynthesis, or a proxy for coral bleaching. And on the x-axis, you have DHW, which is a measurement of accumulated heat stress. And so what I just want to show you here, each one of these panels is a different coral. But if you look at this guy, for example, this line never really declines, even as we move further and further into a more stressful environment. Whereas this one kind of drops off a cliff, right? So we can establish using this technique that this coral is more thermally tolerant than this guy. And this gives us a much more kind of refined and nuanced way to say these corals are stronger than others without relying on just using our eyes to detect bleaching, for example. And so what we pull out of this is an effective dose that's in degree heating weeks, but that's important because that metric is translatable. We expect to be able to compare that to field observations of bleaching events in other parts of the world. And it also frees us from being constrained to the specific kind of profile we use for a stress test. We can compare those results to theoretically people elsewhere who are heating up corals and measuring the same type of thing using a different system, maybe inside, maybe a shorter experiment, et cetera. We're trying to generate really generalizable data points. After we collected this initial data that I just described, these corals got put onto these custom HDPE plates that we have. This is a stainless steel table that's at Reef 13 in Kinohi Bay. And our to stay, and 2 plates destined to be transplanted. And then in May, we actually made those transplants. So you can see on the left, corals in Kinohi Bay just stayed there. On the right, the ones moved to Ulupa'u were installed directly onto the substrate with this kind of nifty 2-part epoxy eye bolt screw process that we came up with. Kira installed these, did an amazing job. They're still there. And this, again, is a step towards more reproducible ways to move corals around and understand how the traits are behaving in different environments. So a few quick results. On the y-axis here, you've just got proportion of fragments, and X is the 2 species. We've got Kinohi Bay on the left and Ulupa'u on the right. And what you can see on the left-hand side here is the corals in Kinohi Bay, generally pretty happy. We only have a little bit of mortality and a little bit of corals that went missing. So as expected, this is a pretty good environment for these corals. Conversely, the corals that were transplanted to Kailua Bay have higher mortality. And I wouldn't say that it's catastrophic, but only about half of those corals have made it the first 3 or 4 months. And this is our first kind of signal that, individual corals do care about the exact habitat from which they came. And I haven't parsed this based on origin yet, but either way, we are seeing this side effect. One thing we are glad about is the fact that this yellow bar here from missing corals is not substantially larger in this much higher wave energy site, right? We're using these little plugs with thumb-sized corals on them. Big waves come through. We're expecting some of those to snap off and be lost, but we're seeing that that's not really happening, which again is important for methods development and techniques for how we might actually be able to outplant these corals. So an initial signal that staying in your home site keeps corals a little bit happier. When we look at these initial thermal performance outcomes, there's a couple of other interesting things happening. On the left-hand side here, you've got your performance on the y-axis. That's just that curve that I just showed you. And you can see that higher values are better. Higher values are stronger corals. And you can see that the Porites, the blue bars, are higher than the pink bars, which are Montipora. So we've got a big species-specific difference. But importantly, we don't have a difference based on where the corals are actually collected from. This is a little bit unexpected to us because we kind of initiated this experiment with the idea that using some of our collaborators' previous results, these corals from Kinohi Bay would be a little more sturdy, would be a little more thermally tolerant. But we don't see that so much at the beginning. I also described the idea that the individual reef being warmer or cooler or average might influence the corals that come from there. The idea there is if one reef, even if it's only 500 meters away from another, is routinely 3/4 or half a degree hotter, maybe that environment has selected for corals that are more thermally tolerant there too. And so what we see over here is not that. We've got Porites and Montipora, the 3 different reefs. And there's a really strong effect of the site that we selected them from, but it doesn't correspond to that idea of local adaptation. So, for example, both the cool and the warm site are really thermally tolerant, while the average site for this species is not. What this means probably is that the corals that are living in an individual place are really driving this effect, and perhaps the environment is selecting for them, but not in the way that we thought. And so it kind of introduces this whole additional layer of complexity to all of these outcomes that we're trying to measure. This was all preliminary data from before we outplanted these corals. And one of the key parts here, next steps at the bottom, is we'll be re-assaying these same corals after they have lived at a different site or at their home site for over a year, and trying to understand if these phenotypes are retained. There's a number of other ongoing analyses. I mentioned Ali Klein again is working on a lot of this stuff. Is thermal tolerance retained post-transplantation, which means, can we identify really strong corals and use them for conservation? How do some of the molecular characteristics like gene expression and biochemistry change? What are the symbionts doing here? And how is the growth impacted by thermal tolerance and transplantation stress? That last part is particularly important, again, because coral growth leads to structure, which ultimately creates the function of a healthy coral reef. So back to Kira.
All right, so while we think the science is super exciting, I think where we had most of our personal growth with this project was with what we learned from the community. So I wanted to cycle back to this for the end of our talk. A lot of this is in a document called Kulana Noi'i, which is provided by the Sea Grant. So definitely check that out. And there are a lot of the same things that I saw, and with referencing that document, but there are some other new things I added as well. So again, this has been 4 years of building this relationship, and we're not done. We want to continue and really deepen this relationship with Waimanalo. He said it's a tough community, but they've had to in order to have their voice heard. And also they have the biggest hearts of any people I know, and they really care about their people and their keiki. So definitely build Pilina before seeking funding. We wish we could have done that. And if you're interested in working on a site, start making relationships right away. I also wanted to share this just because I think a lot of the people in the crowd are researchers. And as we move forward, we want to make sure that we are involving the community in our research and requesting permission first, which is coming up. And when you build that Pilina right away, you can really find out what those community interests are and build your research around what they want to know, and then use their mana'o or what they know to be able to support that research. Including lineal descendants is really important a lot, and especially in a lot of these places like Kailua, where there's not as many Hawaiians that are readily accessible, which is why we talked with the Ko'olapoko Hawaiian Civic Club for some of that approval. It's really about investing that time. And what I really thought helped a lot was when I started to bring my ohana to a lot of these activities. So one of the people that was the first one to say no to the project, she told me just last week, it's really fun to be able to watch your kids grow up. So like building that Pilina and then them getting to know not just me as a researcher, but me as a mom and all these different sides of me personally is really important. Ask permission and respect it when people say no. And if you have an advocate, or if you feel that there's a way that you can move forward still, then make that pivot and take the time to invest in what the community interests are. Incorporate Indigenous knowledge. Waimanalo Puna Research Hui and Kekulenui o Waimanalo in general, they have an aloha circle every time we meet, where everyone introduces themselves. They either oli or pule. All of this is really important, and you see Jazz and Pua and Spencer going into the water. What I love that Pua and Jazz brought into this project is before going out in the site, they had a pule, and Pua shared the mo'olelo of that specific area. So it's really, really deeply important to recognize the place. Co-develop these projects. Don't just come in and say what you want to do. Make sure you're transparent with data ownership and share your budget, and give back to the community beyond the project. So participate in work days, provide gifts or makana, give them funding to be able to do the work that they need to do. When you interview community members, that's their time. So make sure you're compensating them for that. Especially, I had this conversation with Waimanalo fellow research committee members, I was like, do you guys get paid for this? And they're like, nope, we've never gotten paid for this. And this takes up all of their time. There's weekly meetings, there's monthly meetings that they're taking up their time for. Food is really important when working with the community. Make sure you include that in your funding request, especially if you're working with the federal government. And the biggest thing that I learned from Waimanalo is what they really want is to train their keiki. To them, that's the most important thing. If they get their keiki trained, then they can grow up and they can be the leaders doing, both Western science and working with indigenous knowledge and combining those. So with that, I'd just like to thank PI-CASC because they've been such a flexible funder. Not every funder would be like, sure, yeah, go ahead and like figure out what this turns out to be. So that's really, really important. And then also DARPA has come in and funded Ali Klein, who's online, that helped to do a lot of the transplant experiment. So with that, we'll take any questions. Thank you.
Big mahalo to the whole team for being here and sharing with us today. Let's take a question from the room and then a question online. If we have any, let's start here in the room.
You guys gotta come up. Oh, I was just telling them to come up.
There was a huge swell event in Kailua Bay about a month ago. How did that impact your outplanting? And also there's a discharge pipe nearby. Do you see any influence from that?
I can tell you when Ali and I put out those coral plates with a couple other DARPA staff, we didn't know what we were going to come back to because we knew about the high wave energy there. Like we didn't even know if those plates were going to last. And luckily they have, and they held. So Ali we were just out there retrieving them. I think it was after that event. I'm not sure.
When was about a month ago?
I think they're okay. But yeah, we'll check on them. I'm going actually, yeah. So if it's a month ago, we're going back out later this month because we do monthly checks on survivorship if we can get out there because it's so rough and getting around the peninsula is really difficult. So yeah, we'll see. The other part of that question was the sewage.
So I think we should be able to pick up some indirect signatures of that outfall if they're impacting those corals primarily through the symbiont community, essentially density changes. But I don't think we really know what's going to happen. One of the focal points of this project was not evaluating many individual sites so much as looking at this contrast across the environmental gradient. And so a weakness of a transplant experiment with two locations is that we can't really determine a lot of the cause. And so if the corals in Kailua are suffering, we may not know exactly why. I didn't want to say to your earlier point about the wave energy, though, if we want to do this type of work, the Kailua outplant site is more representative of what reefs in Hawaii look like, and we need to figure out the logistics and the actual mechanism by which we can successfully move corals around. And so if we lose them, that'll be a learning experience, and then we'll iterate and try to come up with a solution. But Kinohi is nicely protected in that regard.
I'll just add that there are living corals there. They're more sparse than Kinohi Bay. So, having an experiment like this is one thing where you might lose corals, but we know that corals can survive in that area even with the sewage outfall. So that's why we're doing this transplant now before making a hybrid natural artificial reef out there, because that reef will protect, that'll stop the wave energy, right? And then we'd help kind of find that. And the plan is to outplant behind that where it's more of a sheltered space. So it's really the testing of that makes it a little bit more complicated.
Great. I see a question online. Let's take a question from there. From Elliot Parsons, says mahalo. First of all, there's a number of things in the chat here. Elliot says, I was curious how long it takes to establish coral fragments in a new environment and how long you will need to wait until you can really compare across sites.
That's a great question. I think one of the major limitations of people transplanting corals anywhere in the world has been how long they have followed them. That remains a somewhat open question. I think by virtue of having these repeated measurement intervals. We're really setting ourselves up for success, though, so we can understand if the patterns we see occur quickly or continuously over time. Kira showed a picture of a coral early on in her slides that we had outplanted at a different site in Kinohi that has kind of fully attached itself to the reef after about 6 months. And so this is a nice example of a coral that is fully grown over the plug, and many of the other fragments there have skirted out onto the substrate in an area covering, 5 or 10 centimeters across. So I would consider those to be part of the reef at that point. Yeah, time dynamics for all this stuff are a really complicated part of it.
Do you see another question in the chat? I just want to give our room a chance. Yeah, one more question in the room.
I think I saw you mention that you're interested in measuring how symbionts, the relationship between symbionts and the thermal stress in the coral. Can you talk about more about how you're gonna measure that?
Sure. So the question was about what our plans are for looking at the symbiont community in these corals. First of all, we use 2 species. Montipora capitata has symbionts in 2 different genera, one of which is known to be kind of canonically thermally tolerant, the other which is not. And then Parietes compressa has this very constrained symbiosis. And so many of the effects we see in that species we expect to be due more to the animal. And so we'll be focusing on Montipora for that. We're using ITS2 sequencing, and the metabolomics also picks up a lot of signals of the kind of lipid membranes of the symbionts. And we'll be looking for shifts in the community, decreases in the abundance based on the light regime, and some of the signals of how both the identity and composition of the symbiont is supporting kind of the nutrient exchange with the host animal. But the metabolomics and ITS2 sequencing are the primary way we're going to look at that. Did that answer your question?
Yeah.
All right. One more question in the online audience here from Erica Gallerani. Super interesting presentation. I'm curious about the compensation for community members for their time. Is this something that you incorporated into funding applications? I wonder how you suggest best going about this if trying to build pilina before applying for funding.
Yeah, that's a great question. So we typically do stipends, and that's pretty much just a training payment. That's like a one-time or once a month thing that you can give. It's not hourly or anything like that. But the struggle we had with the stipends is that if that person was employed in another way by the University of Hawaii, then, or RCH, they weren't allowed to receive stipends. So even though this is their weekend time and their nighttime hours, there is conflict there. So we are definitely still learning about the best way. And if other people have advice, definitely happy to hear it.
Okay, I think we'll have to end it there. One more round of applause for our team. I would like to invite everybody to join us for our next Slice of PI-CASC Seminar, which will be on Tuesday, December 10th at noon here in Hawaii. We'll have Dr. Melissa Price from UH Manoa Department of Environmental Management talking with us about comparison of cost effectiveness of conservation actions for endangered species. So please join us for that next talk. Okay, well, thank you for those of you online, and thanks everybody here in the room. I believe we do have a pie, as we often do, going with our Slice of PI-CASC theme provided by Rachel. So thank you, Rachel. And again, well, everybody, thanks for being here.